Ions bind with protons, removing protons from the cell, then the pH is too low. Okay, so conversely, if the pH is too high, you can remove bicarbonate by this chlorine antiporter system. So here chlorine is the driving gradient. And there, since bicarbonate essentially is formed from hydroxide ions, we're removing hydroxide ions. So that means we have now excess protons available, so the pH goes down. So you see that with these three antiport systems that each have their own pH optimum, we can create the appropriate pH. So microphone still good? Yes, still good. I don't think I would ask you to draw this system, but I think it's fair for me at the exam to ask you a question about how this system makes sense. Similarly, right here, I would really like to ask you something like, if I have this ion moving in, is that favored by the proton gradient? Is that favored by the membrane potential? And you can tell me. Okay, and then finally, the last 15 minutes today, we spent on two epithelial systems. So first of all, what is an epithelium? An epithelium is a layer of cells that separates the internal space of the body, where the bloodstream is, from the outside environment, like the gut lumen, or you can also think of the space within the lungs. And even our skin is an epithelial type tissue. So what do we have here? We have the body cavities, if you want, or let's put it differently. But this is where the bloodstream is. And on the top there, we would have the space, for instance, in the lumen of the gut. So this is our digestive tract. That's the outside world for the body. And this is where your bloodstream and your lymph fluids are. It's the inside of your body. And we have this cell layer that separates these two spaces. It's chemical separation. Now we want the transport to be controlled. We want our digestive tract to take up molecules that we need and to not take up molecules that we don't want. So how can we control that? By transporters. So we have transporters that sit in this cell surface here, in this membrane, in contact with the lumen. And this membrane surface, we call it the apical membrane. The apical membrane. Then we have the other side of the cell, where everything is in contact with the fluids, the lymph and the blood in the body, and we call that membrane the basolateral membrane. Really the lateral membrane, where the epithelial cells are close to each other, and the basal membrane, where we're connected to extracellular matrix. This is just the glue on which this cell layer is sitting. So we have a separating cell layer here, one cell, other cell, on and on. Now we need to prevent any movement of water and solutes between the cells. So there is a structure that connects the different epithelial cells, and it seals the space that is outside of the body off from the space inside the body. And we call that structure the tight junction. So tight junction separates the apical membrane domain from the basolateral membrane domain, and it's a diffusion barrier for solutes, which makes that every transport of solutes and water has to go through the epithelial cells. First through the apical membrane, and then through the basolateral membrane. So you should understand this general structure of an epithelium. Now we know about epithelia from light microscopy and from EM microscopy. For instance, here we see the structure of that tight junction. Here we have one epithelial cell, second epithelial cell. This is the extracellular space, the lumen. We would have diffusion of molecules here, if not for the tight junctions. The tight junctions block this transport pathway. There are connections of transmembrane proteins that connect two cells closely together. This is sort of a 3D view of the tight junction. Here we're at the apical side. This is the basolateral side at the bottom. In green we have the tight junctions. This is one cell membrane, another cell membrane. On the right is a cell, on the left is a cell. Extracellular space in between. The tight junction prevents any diffusion here. Alright, now I love the tight junction and its structure. So here we have yet another EN picture. Found it somewhere, doesn't matter where. Here we have the extracellular space. The cell membrane there is highly expanded to create a lot of surface for uptake. So this is the apical side, cell membrane, we go on and on. Here's a tight junction, and then down here we get to the basolateral membrane. There are other connections. This adherence junction makes that these two cells are physically connected. They're anchored together. The tight junction doesn't prevent the shearing of the epithelial layer. It just is giving that sealing effect. So another way to look at the tight junction is in freeze fracturing. So in freeze fracturing, you freeze a cell sample and then you break, remember? And you break through the membranes preferentially, but sometimes you switch over. So we can see here that this is a shadowed image, and if we look at the very small particles, like the one where I have my arrow at, that has to be protein that sticks out. So if the shadowing, here we are, if the shadowing on that small particle is coming from the left, that means that the black, the dark shadowing metal was deposited from that side. That means if I look here, and this is a valley, where this is in the shaded side and this is exposed to our shadowing source. So based on that, we can see here we are in one cell, and here we go into the tight junction. This is actually its structure. It's a whole network of proteins that run from one cell below to the cell that lies on top of it, sealing off any transport in between. So here we're below and here we're in the cell on top. I love the picture of the tight junction. Okay, so now how does this system work in biology? So we have a pH of 1 in our stomach. And you can ask the question, why? Well, this is good for two purposes. One is that many nasty microbes do not survive the pH of 1. So if you eat a lunch that is not so fresh, you will not always get diarrhea. Because very often, the pH 1 in your stomach takes care of this. In addition, that low pH is very good for digestive enzymes for hydrolytic reactions. Hydrolytic reactions are favored at low pH. So we need a low pH in our stomach. How do we get there? We have cells that are called the parietal cells, and they have the mechanism. So here we have the apical side, the basal side, the cell membrane, the basal membrane, and here we have the apical membrane. We get a very low pH. How do we get there? A proton pump. Water splits, it happens, in hydroxide ions and protons. At the expense of ATP, the single proton is pumped out. Now, we are pumping a charge, and we are pumping against steep concentration, because we have pH 7.2 or something inside and pH 1 outside. That's a 10 to the 5th, what in our 10 to the 6th, so over 1,000,000-fold concentration to overcome. That's very steep. That's difficult for the pump. To make it easier, potassium goes in exchange. So the pump is essentially charged neutral. If the pump would pump against charge and against concentration, it would not get to this low pH. But we get potassium in exchange. So we can acidify the lumen, but pretty soon the cell is stuffed with potassium. So you need a pathway for potassium to get out. Therefore there's a channel. So there's a channel. Okay. Now, okay, that's all nice. Water was split, we removed the protons, but pretty soon the cell is then stuffed with hydroxide ions. Can't have that, then the pH becomes very low. So you need to get rid of that. How do you do that? CO2 is a gas, it fuses in. There's enough CO2 around. It combines with hydroxide ions to form bicarbonate. This is actually catalyzed by an enzyme that we call carbonic anhydrase. But even the uncatalyzed reaction occurs at appreciable rates. Bicarbonate then has to be removed, so to get rid of the hydroxide ions, right? That bicarbonate is removed, and that removal is by an antiporter. That antiporter is in exchange with chlorine. So chlorine moves here down gradient into the cell, and chlorine then would accumulate in the cell. We can't have that, so there has to be a channel so it gets out, and chlorine is getting out, and that movement is strongly favored by the membrane potential. So you get hydrogen chloride equivalent to pH of 1, so at a 100 millimolar concentration, 0.1 molar. That's this system. So you need to be able, on the exam, if I give you the drawing of the cell, to fill in all the 4 transporters, there's 4 transporters, that are required to control the pH of the stomach. And for each transporter, you need to tell me how it's driven. So here we're driven by ATP. This potassium channel, I think, is driven by the potassium gradient. It's not favored by the membrane potential because it's positive out. The chlorine channel is favored by the membrane potential, not by chlorine concentrations, because chlorine accumulates here. And this transporter is driven by the chlorine concentration. And probably, since if this pump is working, since bicarbonate would accumulate, also bicarbonate concentration would favor this transport process. The overall transporter is charge neutral, is therefore not touched by the membrane potential. That's that one. You need to know this one. Or I can ask you this guy. How do we take up glucose in our small intestine? So here again we have an epithelial cell, we have a tight junction, we have an apical surface, and we have here relevant in the background the sodium-potassium pump. The sodium-potassium pump was also working in that other cell, controlling the pH, but it wasn't directly responsible for the action of any of the transport systems there. But here it is. So we have a sodium-potassium driving gradient, driven by ATP, remember that. So that means inside the cell has a low sodium concentration. So now glucose can be taken up from the gut lumen. You've had your lunch, digestive enzymes break down the starch to form glucose. You take up that glucose in your small intestine. 2 sodium go in with every glucose. We'll see later why it is 2 sodium. For now, I want you to remember it. Remember, glucose is accumulating to a high concentration here. Actually, there's a pretty high concentration of glucose in the cell, something like 10,000 times higher than there is in the lumen. We need that glucose to accumulate in the cell because there's another transporter that takes it out down gradient. But let's first talk about this transporter here. So glucose moves steeply up gradient. That transport is made possible by the co-transport, the syn port with 2 sodium ions. The sodium is favored both by concentration and by membrane potential because it's positive outside, always. Okay, so now we have a lot of glucose inside the cell. Glucose can get out by a simple uniporter going down gradient. That's how you get glucose into your blood. Now, we're missing one transporter because the sodium that's led into the cell here gets out there, but potassium gets back in exchange. Pretty soon the cell is stuffed with potassium. We need to get potassium out. Therefore, there's a channel. There's a potassium channel. I'm drawing it for lack of space in the next cell, but it's in, of course, in all of these cells. It lets the potassium out. So, now what we get, net we're moving sodium and glucose across the epithelium, and then water is going to follow by osmosis. So this is actually the mechanism by which you hydrate your body. You're taking up sodium and nutrients like glucose or amino acids at the same time by osmosis. So you will not forget this, because I'll tell you a story. 150 years ago, the 49ers and other pioneers were coming west, right? And they had their wagons and their mules. Well, guess what? What they forgot to put on their wagon was a refrigerator. So, they had their guns, and many people died, not because of wars with Indians and other cowboys, but because they every now and then shot something. And they didn't have a refrigerator, so they carried it with them, and after a couple of days it wasn't so fresh. So they got disease in their digestive tract, diarrhea. Bacteria causing them to secrete water. So many people got fevers and got diarrhea and died of dehydration. So number one cause among the pioneers of death. Had they taken cell biology, they would have known that if they would have just taken a little bit of water or tea, put in some glucose and a pinch of salt, then very easily they would rehydrate themselves. Because you need to have both sodium and glucose at the same time in order to get this process to work. The reason is, it's a symporter. Sodium glucose symporter. And the rest of the system follows. Okay. We'll do a little bit of a review on Wednesday. This was